All right, welcome everyone. Today we're going to be talking about sequence alignment and dynamic programming. So here we are, we're starting the first module of the course, and it's going to be all about dynamic programming and then Bayesian inference and how do we use HMMs and rapid string search. So the module 1 is about genomes. So the computational foundations for these modules are going to be dynamic programming, and specifically, how do we see exponential places, exponential spaces? How do we explore exponential spaces in only polynomial time? Which is sounding a little magical, and I want you to sort of really appreciate the awesomeness of it. So we'll be emphasizing just how hard and how big these spaces are. We're also, next week, are going to introduce hidden Markov models, which is a central tool in computer science. And we're going to look at hidden Markov model algorithms like decoding, evaluation, parsing, likelihood, and scoring. And we're also going to look at hashing, which is, again, hugely pervasive in computer science, how to do content-based indexing. So this week, we're going to be focusing on sequence alignment and comparative genomics. We're going to look at local alignment, global alignment, and how to infer nucleotide-level evolutionary events. And next lecture, we're going to look at database search. How do we scan for regions that may have common ancestry super blazingly fast? Right now, we're going to be looking at how it's an exponential problem we're trying to solve and how we're going to solve it in polynomial time. And then next week, we're going to make it actually linear time, which is crazy. And then we're going to look at how we can model genomes with HMMs. Okay, so that's module 1. To start with, if you look around you, the whole world is populated by moving things that are either attacking us and trying to kill us, like the coronavirus, or helping us digest our food, like the about 30 trillion cells of bacteria that are inhabiting your body, or they're part of you, and like the other 30 trillion cells that are actually human cells that you carry along with your bacteria. Like the yeast that we use to make our bread and our wine, like the fruit flies that go onto your wine if you let it sit too long, like the first, organisms that actually had a spinal cord with central processing units, like the Drosophila and mosquitoes that are way deadlier than COVID. So malaria, for example, kills millions of people every year. Like the first vertebrate genomes and all of our vertebrate relatives, including our cute little fuzzy friends that are helping for biomedical research, of course, and our cousins that have actually a lot of traits just like us, except their forehead stops at their eyebrows and ours continues with this massive brain right above it. So all of life, all of these incredible diversity that you see out the window, below the sea, if you go diving and snorkeling, and in the air, all of these things evolved from a common ancestor. And this common ancestor gave rise to many lineages, some of which, as you know, were extinct. So, there was a giant asteroid impact in Chicxulub, just north of Chichen Itza in the Yucatan, that basically killed off the vast majority of dinosaurs, except for some that evolved to become crocodiles and some that evolved to become birds. And extinction is part of life, sorry to say. And that allows us to start studying the evolutionary relationships between all these different species. So what we can do is actually take genomes of closely related species. Here's a bunch of different species of Drosophila. My uncle, Michael Cambiselis, was actually one of the researchers in Hawaii who was actually studying these Drosophila species, which are just amazingly interesting and intricate. But, everybody says, hey, well, why is the genome of the human so superior to those of flies? Well, it turns out we can't fly. We don't have 100 eyes. We can't move at millisecond speeds. So, flies have a lot of advantages on humans, but they basically have, very comparable genomes, very comparable numbers of genes. And if you look at the genomes of more and more closely related species, what you see is that the genes of these species are roughly in the same order and roughly in the same orientation. So here you can see the genes encoded on the Watson strand, which is pointing this way as yellow, and genes encoded on the Crick strand, which is pointing the other way as, some kind of pink, purple. And then you can see that their order is roughly preserved based on these lines that sort of show you the matching genomic coordinates across different regions of the genomes for those species. And these kinds of alignments span over hundreds of genes at a time, which are basically collinear. So what you realize is that all of these species are basically different versions of each other. It's like, I don't know, one is running Windows 98 and the other one is running Windows 2000. And they're all sort of just slightly different, but more or less the same functions are preserved. And that's very important because we can use comparative genomics to recognize functional elements. We can basically compare Windows 98 and Windows 2000 and look for instructions that have been preserved. And those instructions are more likely to be functional, whereas a lot of random bits of information may have been flipped that are non-functional. So to make it much more concrete, if you look at the human genome, the vast majority of the human genome does not code for protein. So only 1.5% of the human genome actually codes for amino acids that make up the 20,000 genes that make up a human, genome and a human gene set, and ultimately all of the protein functions of the human genome are encoded in just 1.5% of your genome. This is mind-boggling. The other 98.5% do not code for protein. And if you look across closely related species, like dog, mouse, rat, you see that this other 98.5% is largely non-conserved. So the vast majority of the human genome appears to be evolving neutrally. Only about, I don't know, 5 to maybe 15% of the genome is clearly recognizable as conserved. So that can basically help us pinpoint where are the functional elements within these genomes. So we can basically look within the conserved regions and basically say, well, maybe conserved regions can help us find genes. Maybe they can also help us find other stuff that might be, I don't know, gene regulatory or playing control functions to control these genes. So we can use comparative genomics, which basically means lining up the genomes of different species to reveal functional elements. For example, protein coding exons, these are the parts that are kept when splicing happens. Protein coding exons are deeply conserved to mouse, chicken, fish, etc. You can see that every time there's a protein coding exon in dark blue here, you see that it is conserved all the way to all of our vertebrate relatives. By contrast, there's many other elements that are also strongly conserved. And the question is, are they exons, are they regulatory, and so forth. So what we're going to look at when we get to the evolution part of the class, which is actually a little later, we're going to be developing methods for estimating the level of constraint. We're going to be counting the number of edit operations, the number of substitutions, and the number of gaps as a measure of constraint. We're going to be estimating the number of mutations, including back mutations, using probabilistic models. We're going to be incorporating information about the neighborhood and looking for conservation windows using HMMs. We're going to estimate probabilities of constrained hidden states. And in fact, that's what I'm showing you here. So for those of you who've heard of HMMs, which we're going to cover in 2 lectures, hidden Markov models basically allow you to estimate the hidden state based on the observed variables. And then the observed variables here are the amount of conservation that you see. And then the hidden state has a posterior probability of being a conserved state or a non-conserved state. And then that probability is higher for those regions that are truly conserved. So that's another way that you can estimate constraint. Or you can actually use the phylogenetic relationships relating these species in a tree to basically understand how many substitutions happen along the tree. Okay, so let's see who's with me so far. And I'm going to be reading the numbers because I'm erasing them every time I do a poll. So if you hear me say a string of 5 numbers, it's basically number of people following above 80, 60 to 80, 40 to 60, 20 to 40, 0 to 40, or 0 to 20, and so on. So here's the numbers, 28, 10, 0, 0, 0. This is awesome. And then the other question that I have for you guys is, how's the pace? Am I going too fast or too slow or just right? Oh, good. Cool. So as for the pace, it's 28 on the dot, 5 too fast, 4 too slow. There seems to be a chat question. If you have the full genetic data, what hidden states would you have? That's such a great question, Arez. So the question is, why do you need a hidden state to estimate? And the answer is, what you have is observation of whether something is conserved or not. What you're inferring is whether that region is under constraint by evolution, because it might appear conserved just by chance. Based on the amount of evidence you have, you're able to sort of weigh that probability. Arez, does that answer your question? It's a great question. So basically, the hidden state is it truly functional or is it not functional? But the observation is, what is the number of nucleotides that are conserved? And Lily is asking, what is a functional gene? So functional means that it matters. If you break it, the organism will probably have a disadvantage. It might not die right away. That gene might only be helpful every 17 years when there's a giant, I don't know, snowstorm. And over evolutionary time, you know, for the first 16 years, it might be fine. And on the 17th year, all the people who had the faulty version of that gene might just die off, all of the organisms that have that faulty version. So that's how evolution works. It basically, you know, sums over huge evolutionary spans to basically measure function that way. And Manu is asking, what do the many genes that don't encode for proteins do? So there's about 20,000 genes in the human genome, and they encode for proteins. We know the function of maybe 80% of them. The other 20%, believe it or not, we actually don't know what they do, and it's quite remarkable. But in addition to the 20,000 protein coding genes, there's about 2,000 long non-coding RNAs, and many of those act as scaffolds for chromatin to fold. Others are just simply not hurting, and therefore they're just not really functional, but they're being transcribed. Perhaps the act of transcribing them is itself functional, and perhaps they have function we don't yet understand. They're usually expressed at very low abundance, so it's very hard to find their function. And then in addition to the long non-coding RNAs, there's a gazillion short non-coding RNAs. Those can be tRNAs that help translate mRNAs into proteins. These can be splicing RNAs. These can be ribosomal RNAs that help translate. I hope that answers your questions. All right, so one more question. Who's finding the questions helpful? So give me a 5 if you find the questions helpful, 4 if you're so-so, 3, 2, 1 otherwise. So great. So for those of you guys who are asking questions, these should be some positive reinforcement, because 17 people said super excited about the questions, 13 said yay, and 7 were in the middle, and only 1 person was at the bottom. So it's, you know, it's important to ask questions, so keep them coming. All right, so that's the basis of why we need comparative genomics. But everything I've been telling you is assuming that we can kind of actually align genomes. So today, we're going to be learning, how do we actually align genomes? How do we create these alignments based on which we can then study the evolutionary signatures of different classes of elements? We can recognize the specific patterns of evolution of protein coding genes versus non-coding regions, the specific patterns of evolution of RNA structures, the specific patterns of evolution of microRNAs and regulatory motifs and individual motif instances. So these are particular classes of elements that we can distinguish, not just based on their level of conservation, but based on their pattern of evolution. And we're going to be learning more on that in lecture 13 or 17 or something like that. But today, we're going to be focusing on how do we even generate these alignments? How do we take one sequence from one species and lining up with a sequence from another species, okay? And this is actually, you know, very near and dear to my heart, because that's a slide from my PhD thesis. So if you search for, I don't know, Kellis Nature 03.pdf, you will find that, yeah, a long, long time ago in a galaxy far, far away, I was a student too. And this was my first paper. So Manolis, Kellis. And if you look at figure something or other, ta-da, figure 6 of this paper, I am so proud to now be teaching it in a lecture. So this is kind of cool. So anyway, so here's an alignment of the yeast Saccharomyces cerevisiae. I'm Greek, so I'm going to be giving you etymology for a lot of stuff. So Saccharomyces means zacharo, which is sugar, and then mycis, which is mucus, which is fungus. So sugar fungus, and then cerevisiae is the same as cerveza, so sugar fungus that makes beer. So the sugar fungus that makes beer, also known as baker's yeast, much more PC, aligned with Saccharomyces paradoxus, Saccharomyces micatai, and Saccharomyces baianus. What we're showing here is a multiple sequence alignment that wraps around multiple lines, and then a star denotes all of the places where that nucleotide was perfectly conserved in the alignment that we generated. And what's really exciting about this is that if you look at the Tata motif, it is perfectly conserved, both for this gene, GAL1, and for that gene, GAL10. If you look at the GAL4 motif, it is perfectly conserved. So 3, 11, 3. This is one turn of the double helix. If you look at the MIG1 motif, it is quite well conserved. And then there's a second MIG1 motif, which is not super well conserved. And then there's a second island of conservation, which is not matching motifs. So why is this exciting? Because it means that we can actually read evolution just by staring at alignments. And that basically also means that we can use those alignments to actually decode functional elements. We can basically say, well, where are the islands of conservation? There must be something important there, because all these other regions are not conserved, but those islands are conserved. So that probably means that they're important. So we can read evolution to reveal functional elements, okay? So our goal today is to make that possible, to actually align two genes. And the way that we're gonna do that is that we're gonna first formulate the problem. So, and the reason why I'm going a little slower on this is because this is the kind of exercise that all of you guys will be doing in your project. So it's very important to see it in practice. So we're going to take a biological question, translate it into computational terms in different ways, and we're going to look at different formulations, and then we're going to see how all of these formulations are in fact pretty hard, how there's in fact an exponential number of possible alignments. And then we're going to introduce the computational technique that we're going to be using to solve these exponential number of problems, and then apply that computational technique directly to sequence alignment. So first we're going to introduce the problem, then we're going to introduce the technique, and then we're going to combine the two. And then lastly, we're going to look at some cool advanced topics for variants of that end product. Okay, so what we've seen so far is comparative genomics and molecular evolution, and just a brief introduction to why evolution is there and how it works, and just sequence alignment. And now we're going to look, how do we translate this biological problem into computer science terms? So first of all, genomes change over time. There's mutations, there's deletions, and there's insertions. And these just happen because as DNA polymerase is happily walking along and and copying DNA into DNA, it's basically opening up the double helix, and then it's making a second copy here, and it's making a second copy here. So, you know, this semi-conservative replication of DNA, where you have the old strand and the new strand on one side, and then the old strand and the new strand on the other side, every now and then makes errors. It basically sometimes inserts the wrong nucleotide, and sometimes it inserts a gap, and so on and so forth. So these things just happen, okay? And it sounds like mistakes, but the mutations are also the agent, they're the power force of evolution. I always like to say that if we didn't have mutations, if engineers had designed evolution and the copying machinery, then we would be perfectly replicating bacteria. We would have just never had mutations that we evolved with. And basically, mutations are both detrimental, but they're also the agent of evolution. They also allow diversity that sort of selection then keeps or rejects. So these are the basic operations of evolution. And the question is, if all you have is the starting sequence, namely Saccharomyces cerevisiae, and the ending sequence, namely Saccharomyces paradoxus, then how do you transform one into the other? How do you infer the series of mutation, deletion, and insertion operations that gave rise from one to the other? Okay. So that's the part of formalizing the problem. So in fact, there's a question. Let's see. Does that mean that the gene length is not conserved over a short time span? No, gene length is not an issue. As long as it sort of maintains the function, you don't need to worry about gene length. So the first thing we do is we define a set of evolutionary operations: insertions, deletions, and mutations. And making these operations symmetric allows for time reversibility. And that's part of our design choice. And the reason for that is that we don't really compare, Saccharomyces cerevisiae to Saccharomyces paradoxus. We don't really compare human to, mouse, where, human arose from mouse. No. We both arose from a common ancestor. And basically, the reason of time reversibility is that as we're aligning human to mouse, we want to be able to sort of talk about operations that are directional. And whether they are moving in one direction or the other direction, it shouldn't matter. And that's why we have this time reversibility. And there are some exceptions with methylated CpG dinucleotides that sort of become unmethylated and lose the methylated Cs that become Ts. But you don't have to worry about that. The next thing we need to ask is, what is our optimality criterion? How do we know we're doing well? Is this a good solution? So maybe what we would like to have is a minimum number of operations. Just how many insertions, deletions, and mutations. Or alternatively, we might want to have minimum cost, where these operations might actually have different costs. And depending on the cost, we might prefer to have 3 cheap operations than 2 very expensive operations. The third is that it might actually be impossible to infer the exact series of operations, because as human transforms into mouse and vice versa, getting this time reversible model, you basically might take a very circuitous path where you change an A into a T, and then back into a C, and then back into an A. It looks like that never changed, but in fact you made several operations that are invisible. But what we're going to choose is a minimum path, a minimum cost transformation. And that's Occam's razor. Basically, Occam's razor says, among multiple hypotheses that explain the data equally well, I'm going to choose with my razor the sharpest hypothesis, the smallest model. So from all of the super highly complex models that I could have that explain the data equally well, I'm going to choose the smallest of these models, the simplest. We're also going to have to, once we have the optimality criteria, we're going to have to design an algorithm that achieves that optimality or that approximates it. And the tractability of that solution will depend on the specific assumptions that we make in the formulation. For example, if we, we might make some decisions that capture every last aspect of the biology. For example, CpG dinucleotide non-reversibility, or this, weird back and forth over time, etc. And these might make it more relevant to biology, more correct. It might handle more special cases, but it will push us away from tractability, computability, implementation, and so on and so forth. So basically, there are trade-offs. We can make some simplifying assumptions that make things more computably easy, or sort of handle some spatial cases that make this harder. And there will be trade-offs in all of your final projects as you're designing this. So, and of course, not all decisions are conflicting. Some decisions might be both more relevant to the biology and more tractable. And there's a famous example of Pevsner versus Sankov arguing about the directionality of chromosomal inversions. Turns out that inserting directionality as a criterion and making it more biological also makes it more tractable and moves it away from NP. All right, so now let's look along this continuum of formulations. What are different formulations that we can use for our evolutionary problem inference? So the first formulation we're going to look at is longest common substring. Given two possibly related strings, S1 and S2, what is the longest common substring without any gaps? So if I have substring one and string one and string two, a substring is a contiguous set of characters. So what is the length of the longest common substring that you see here? Feel free to unmute and shout it out. So I see length 3 with, I don't know, TCA, matches TCA. It's pretty good. And then there's a length 4, which is, GTCA, right? So if I shift my sequence by only one offset, I get TCA. But if I shift it by another offset, I actually get GTCA over here. So this is kind of cool, right? We were able to basically search the two sequences, scan them past each other, and that's a computational challenge that you guys solved in your heads in being able to sort of find these three and then find these four. What is the runtime of that algorithm that you used? Well, at every offset, you probably compared a certain number of characters, and then, you know, it was an n-squared operation because you had a bunch of starting points, and at every starting point, you had to compare every part of the sequence. But many of you probably solved it much more smartly by sort of aligning individual characters and then extending from that alignment. And that's the basis of the BLAST algorithm that we're going to look at on Tuesday. So anyway, that's the basic algorithm for finding longest common substring. But this doesn't handle gaps. Insertions and deletions are not modelable using this. So we need to increase complexity a little bit. So the next formulation is going to be longest common subsequence. And a subsequence is the longest common, not necessarily contiguous set of characters. Notice here that I can capture my TCA that I had before. And if I just insert one gap here, I have G and T. And if I insert another gap here, I also have A. So now I can capture many more characters that have common ancestry because I'm tolerating insertion and deletion events. And you know, this is inferring a gap in one sequence, you know, an insertion of a T in sequence 2, deletion of C and deletion of A in sequence 1, and then mutation of C to G. And if you notice, this is exactly the example that we had earlier. So we basically created the longest common subsequence between this and that, and found an insertion, 2 deletions, and then a mutation, okay? So that's what we would like to do. We would like to solve that original problem that we posed by a cool algorithm that can do that, okay? And again, every such alignment immediately infers and implies and implicates a series of events, 2 deletions, 1 insertion, and a mutation, okay? So longest common subsequence is in fact the same as edit distance, where the number of changes needed between S1 and S2 with a uniform scoring function is in fact what the longest common subsequence actually solves, okay? But we can go a step further. We can start modeling that gaps, yes, might have a fixed penalty, such as every insertion and deletion has a unique cost for every character, or you might actually have varying penalties for different gaps. You might also have varying penalties for different types of operations. So for example, a transition between an A and a G both of which are pretty bulky bases, happens much more easily than a transition, than a change of an A into one of the smaller bases, a T or C, just because of their stoichiometry inside DNA polymerase. These types of mistakes, big to big is replaced much more easily, and small to small much more easily, rather than big to small, which is replaced much more rare. So we might actually want to encode a score matrix rather than penalize every mutation exactly the same amount. We might want to include a score matrix that basically penalizes a mismatch between A and G by only half a penalty, and then penalizes a mismatch between T and C by a full penalty, and then rewards matches with a +1. Okay, so let's see who's with me so far. Awesome. Very good. So 31,5,0,0,0 is pretty good. Okay, so these are increasingly increasingly complex formulations, because polymerase confuses A with G and Z with T more often. So let's encode that. And then another formulation might be to vary the gap cost. Basically, a linear gap could be the same as before. An affine gap might have a big initial cost for starting or ending a gap, and a small incremental cost for each additional character. And that's because polymerase sometimes starts slipping, and when it starts slipping, it might slip by multiple characters, by multiple nucleotides. Or a general cap penalty could basically allow any kind of cost, but that's no longer computable using the same model. You could also build a frame-aware gap penalty, where multiples of 3 that disrupt coding regions, that preserve coding regions are fine, but multiples of 3 that disrupt coding regions, like anything that's not mod 3, that's mod 3.1 or mod 3.2, is in fact disrupting the reading frame of translation, and that could be penalized. We might also want to seek duplicated regions, rearrangements, and we're going to look at all of that when we talk about local alignment on Tuesday. So the question is, how many possible alignments are there? And in fact, there's a question. Let's see. How do you choose which gap penalty to apply? It really depends on your problem at hand. So basically, if you want to go really fast, you might just want to go linear gap. If you want to go really fast but capture additional biology, you go with an affine gap. If you know that you're aligning protein coding regions, you might want to build a frame-aware penalty. So it kind of depends on the application and your expectations. So let's see, how easy is this problem? So, it took us a while to find, the substring. It took us maybe a little longer to find the subsequence. How long does it take for you guys to compare a sequence of 1,000 nucleotides with another sequence of 1,000 nucleotides? So you basically have to scan every single position and then choose at every one of those positions whether you want to insert a gap or not. And then you're making a binary choice in each one of 1,000 positions. So that's basically 2 times 2 times 2 times 2 times 2 choices 1,000 times. So that's 2 to the 1,000 possible alignments. Okay, so if you're trying to align a sequence of length n to a sequence of length m, and you don't want to align a gap to a gap, the longest possible alignment you want to make is n plus m. And this is equivalent to making a choice at every position, each of the n plus m positions, about whether to insert a sequence here, a character here, or a character here, or a character in both. Okay? And then, besides some constants, this is effectively 2 to the n plus m, which is basically a hugely exponential number of possible alignments. Okay, so if I want to enumerate and score every single alignment, I just don't have the option to do that. I need a faster solution for finding the best alignment. And what I need is a polynomial time algorithm for finding the best alignment among an exponential number of possible alignments. So how do I do that? The answer will always be dynamic programming, okay? So let's see. Who's with me so far? So basically talked about comparative genomics and molecular evolution. How do we translate biological problems into computational terms, and why it's hard, why there's an exponential number of possible alignments. So first of all, how's the pace so far? Awesome. So I basically have 28 spot in the middle, and then 5 too fast and 2 too slow, which is great. And then how well are you following so far? Awesome. So 26, 7, 0, 0, 0. That's great. Okay, so now let's dive into it and basically introduce the principles of dynamic programming. And first of all, we're going to introduce a super simple way of doing it, Fibonacci numbers. And then we're going to see how the top-down solution is exponentially slow, and the bottom-up solution is only polynomial in time. And then we're going to study that solution by recognizing that we've detected and eliminated repeated subproblems. We've been able to order the computation in a way that the subproblem is already solved when we need it, and then build a table lookup function that allowed us to look up the operations that we need to compute over and over again, so we don't have to recompute them. And we're going to look at this general recipe for dynamic programming of parameterization, subproblem space, traversal order, recursion formula, and then traceback. And then we're going to turn to apply that to sequence alignment. So how do we turn exponentials into polynomials? Let's start with Fibonacci numbers. So if I want to compute Fibonacci of 6, that's Fibonacci of 5 plus Fibonacci of 4, which Fibonacci of 5 is Fibonacci of 4 plus 3, and Fibonacci of 3 plus 2 is Fibonacci of 4, and so on and so forth. Where do Fibonacci numbers come from? Well, they're everywhere in nature. If you look at a nautilus, and you look at the sizes of the different chambers as the organism grows, you basically see that every year it kind of makes another turn whose length is actually the sum of the previous 2 turns that it had made, okay? So that ends up with sizes of 1, 1, 2, 3, 5, 8, 13, 21, 34, 55, and so on and so forth, which is basically the Fibonacci series, okay? If you look at the spirals of the Romanesque, totally different evolutionary lineage, same thing, Fibonacci numbers are everywhere. Coneflower spirals, leaf orderings, the leaves per height of most trees that you will observe, or Leonardo Pisano Fibonacci's original example, the number of rabbits per generation. So you basically have 2 bunnies the first summer, and then every summer they grow, and then after they've grown, they reproduce, and they make another set of fresh bunnies. And these bunnies grow and reproduce. And then every year thereafter, the bunnies that have come of age continue making more bunnies year after year after year. So that ends up with a series of how many pairs of bunnies do I have? I have 1, 1, and then suddenly I have 2 pairs, 3 pairs, 5 pairs, 8 pairs, 13 pairs, 21 pairs, and so on and so forth. So it grows pretty fast. And that's what, you know, Fibonacci actually wanted to model when he came up with these numbers. So if I wanted to write a Python program that computes Fibonacci numbers, I could just say that Fibonacci of n. If it's 1 or 2, simple, return 1. Otherwise, I'm just going to return Fibonacci of n minus 1 plus Fibonacci of n minus 2. That's basically the definition. So if I want to compute the nth Fibonacci number, it will basically take me n minus 1 plus n minus 2, which if you expand it out, is basically n minus 1 plus n minus 2, which is, you know, n minus 2 plus n minus 3, which is n minus 3 plus n minus 4, and so forth, and so on and so forth. So that analysis is basically expanding out to the sum of, you know, n plus n minus 1 plus 2 times n minus 2, etc., all the way to 1, which is basically 2 to the n. So it's an exponential function. And you can see here the exponential with the depth of n. I need to compute order of 2 to the n leaves, because at every generation, I double the amount of work that I need to do. Okay? With Fibonacci of n, I have 100 doublings. Everybody with me so far? Good. All right, so what's wrong with that? Well, the problem is that if I compute Fibonacci of 3 once, why do I have to compute it again here and here? If I compute Fibonacci of 2 here, why do I need to compute it again here and here and here and here, and so on and so forth? So I'm redoing the same operations over and over again, because Fibonacci of n minus 1 will again recall Fibonacci of n minus 2. But that will be in the computational loop here, which won't even know that I have already done it here, and so on and so forth. Okay? So the correct way of doing this is to basically say, oh, you want Fibonacci of n? Why don't I give you all of the Fibonacci numbers, all the way to n? I'm doing a lot more work, but I'm doing it much, much more cheaply. So instead of giving you just one number, I'm going to produce a table, Fibonacci table, that will basically have at every point the number that this corresponds to. So Fibonacci of I will have basically the I Fibonacci number. So the way that I'm going to compute it now simply has Fibonacci table of 1 plus 2. And then for I in the range of 3 to n plus 1, Fibonacci table of I equals Fibonacci of table I minus 1 plus Fibonacci table of I minus 2. Notice both I minus 1 and I minus 2 are already computed by the time I get to I, because that's how I've ordered the computation. So, and if you look at the runtime, it's just computing Fibonacci of 1, 2, 3, 4, 5, 6, and then it's done. Okay? Who's 100% with me so far? Let's see. Awesome. So what are the ultimate principles that we've made? What we've basically learned is that if we order the computation just right, and by the way, 33,3,0,0. If we order the computation just right, we can make sure that whatever intermediate results need to be computed are already available. What else did we do? We basically said, well, hey, there's only a finite number of subproblems. Let me solve them all, just from the bottom up. Instead of going from the top down, I went from the bottom up. So number one, I built a table. Number two, I organized the computation in a way that all of the results that I needed were available when I needed them. And number three, I basically went from the bottom up rather than from the top down. Okay? So these are basically the principles of dynamic programming. So we reveal the identical subproblems. We order the computation to enable result reuse. We systematically fill in a table of results, and we express larger problems as functions of their subparts. And the ordering of the computation matters. The naive top-down approach was very slow, and the results of the smaller problems were not available. So there was a lot of repeated work. But for the systematic bottom-up approach, we were successful because we systematically solved each subproblem. We filled in the table of subproblem results in order, and we looked up the solutions instead of recomputing them each time. So these are the hallmarks of dynamic programming. Number one, there's optimal substructure. The optimal solution to a problem instance contains within it optimal solutions to the subproblems. Number two, there's overlapping subproblems. There's a limited number of distinct subproblems which are repeated many, many times. And dynamic programming is typically used for optimization problems. So Fibonacci is not really a dynamic programming example because there's no maximum, there's no optimization, but it teaches you all these other principles of ordering the computation. The optimal choice will be made locally by taking a max in these optimization problems. And then the score will be added through the search space. And then at the end, we will have typically a traceback to find an optimal path from the individual choices. And in terms of where it is in the range of difficulty, there are easier problems like greedy choice problems, where a greedy choice is always possible at every step. Dynamic programming actually requires a traceback at the end to find the optimal path, whereas greedy doesn't require it. You just get to the optimal path directly. And there are, of course, harder subproblems where there's no optimal substructure. For example, if there are dependencies between different subproblems. So if in my tree, this subproblem depended on some other kind of subproblem, then this would not be possible. Basically, I would have to sort of skip across different parts of the tree. So again, both greedy and dynamic programming have optimal substruction overlapping subproblems, but for greedy, the greedy choice is possible, but for dynamic programming, you need a traceback to find the globally optimal solution through many local choices that you've made along the way, and you've recorded which direction gave you the best choice. So what you'll typically need to do in practice is find that matrix of parameterization, decide on the number of dimensions, the variables, make sure that the subproblem space is finite, it's not exponential. For example, if not all the subproblems are used, you're better off using memoization or some other technique that just, stores things on the fly. And if reuse is not extensive, perhaps dynamic programming is overkill. Maybe, it's not the right solution. You could just only solve the subproblems that you'll actually need, whereas dynamic programming actually solves all of the subproblems. Also, the traversal order matters greatly. You want to make sure that the sub-results are ready when you need them. And bottom-up is usually the way to go, but it's not always obvious what bottom-up means. And then for the recursion formula, the larger problems are always expressed as a function of the subparts, and finding how to express those will be the art of dynamic programming. So, sort of setting all that up and then deciding how to express these. And then lastly, remembering the choices. That function here of the subparts will typically include a minimum or maximum, and you need some kind of One of the joys of remote teaching is that every now and then I will actually lose my internet connection. So I apologize for that. I hope you didn't miss too much, but all right. So as I was saying, you need to remember your choices, and then you need a representation for storing these pointers. And, you need a polynomial space of solutions that you have to go through. And then once you have those 5 parts, you then start computing. You systematically fill in your table of results, you find your optimal score, and then you trace back from that optimal score to find the optimal solution. Okay? So now that we have the parts in place, let's now apply this to sequence alignment. Okay? But right before, who's with me so far? So let's see. Awesome. So 23,9,0,0,0. Okay, so let's now build dynamic programming for sequence alignment. Remember, what we're trying to do is figure out the series of evolutionary operations that relate one sequence to another sequence. And there's an exponential number of those problems. So we're going to use now dynamic programming in that space to find those. So we're going to look for repeated subproblems, decide on our matrix parameterization to sort of express these subproblems, build a recursion formula, and then remember the choices and then trace back. Okay? So how do we apply dynamic programming to sequence alignment? And by the way, this is awesomely cool. So I hope you guys are excited about that. So the key insight is that the score is additive, that we're going to move from smaller to larger subproblems. Okay? So we're going to compute the best alignment recursively. And the key idea is that the best alignment going through this character C aligned to this character A is going to be the best alignment of that part to that part, plus the best extension from that subalignment. Okay? And if that's not part of the optimal solution, if there's a better alignment, if that part is not the best subalignment, then I get a contradiction because I could just swap that in with a better alignment and then get a better score overall because the score is additive. So that's where the cut and paste argument that you probably have seen in 604.6 comes in to basically prove that the sub-solution must be optimal. Not every optimal sub-solution will be part of the optimal global solution, but every sub-part of the global optimal solution must have within it optimal subparts. Okay? So we're going to be breaking up our sequence by indexing with the ith position of one sequence and the jth position of another sequence. So it's going to be a two-dimensional parameterization, and I can express that very naturally in a two-dimensional grid. So here I'm going to have all of the characters of one sequence, and this character here is the C, is the ith character of the first sequence, and I'm going to be aligning that to every character of the other sequence, and the jth character is going to be, matching to the ith character, and that's going to be the G here. So what I'm going to be asking is, how ever did I get to that alignment of I with J from sequence 1 to sequence 2? That must be optimal, and if I have that optimal sub-alignment, then I can build on it to extend to the optimal alignment overall. So with a single recursion, top left to bottom right, instead of two recursions, middle to outside, I'm able to now fill in a two-dimensional grid that basically tells me what is the score of the sub-alignment all the way to I and the sub-alignment all the way to J, the sub-alignment of S1 all the way to I and S2 all the way to J. What is the score of that sub-alignment? And then if I can reuse that score, I'm golden. So that's what we're going to have to do now. Okay. So now let's see, how do we compute this score? Well, to compute this score, I want to reuse something from smaller scores. So to calculate the alignment of ACGTC versus TAG, I want to find the maximum alignment score. I'm going to choose the maximum among exactly 3 options, because all others go through these options. The first option is going to be, well, I'm going to extend the optimal sub-alignment of ACGT versus TA. And if I have the score of that, I can just extend it by 1 character in S1 and by 1 character in S2. And that gives me whatever that previous score was, and I don't need to know how I computed it. I just need to know that it was optimal. And I've stored it in my matrix. I'm just going to look up that score, and I'm going to extend it by adding 1 more character in each sequence. Who's with me on this one? Okay. Very nice. So 17, 15, 1, 0, 0. So if I have already computed this score, I can just extend it by 1 character each time and pay whatever penalty I have to pay to align C with G. Probably just a mismatch score of aligning C to G. An alternative is that I could instead reuse the score of ACGTC aligned to TA with 1 more character here. And where do I have that score? I have it right above on my matrix. I have it right here. So as I'm computing this one, I can reuse the score from here, extending in both directions, or I can reuse the score from here, adding a gap, or I can reuse the score from here, adding a gap. Okay? So adding a gap here and inserting a character here, that basically pays the gap penalty, and then I just reuse the previous annotation, the previous score from that alignment. Or alternatively, I could insert a gap in that direction and then pay the gap penalty and reuse the score from here. To clarify, the score is a weighted edit distance for the ACGT basis correct. It's a weighted edit distance between ACGT and TAG. So basically, it's the score of that pair, and the score of this pair, and the score of that pair. Every such sub-alignment I can just store in my matrix. So I've stored this one in position 5,3. So I'm entering now the score for position 5,3 by reusing the scores that I had at position 5,2, at position 4,2, and at position 4,3. Okay? So I'm sort of reusing the computation. So the key idea is that we're calculating the maximum alignment score of the longer sequences based on previously computed scores of shorter sequences. We're assuming that all shorter alignments are already computed, and we show that you can use them to compute the longer alignments as a function of them. And by induction, we can compute alignments of any length this way. So if I have anything that's shorter in this direction, shorter in that direction, or shorter in both directions, I can compute the next one. Well, then I'm done. By induction, I can just fill out the matrix from beginning to end. And the implementation is that we're going to store all of these scores in a matrix that has the score for the S1 prefix and the S2 prefix alignment. And we're going to fill the matrix systematically from the bottom left, from the top left to the bottom right. Okay. So these are the 4 entries that we were just considering. 5,3 computed as a function of I minus 1, j minus 1, I, j minus 1, I minus 1, j. Okay. So these 4 quadrants are just entered here in this matrix. And what I'm going to be computing is the score of this matrix from the top all the way down. And then the path through the matrix is actually going to correspond to an alignment of S1 and S2. So as I traverse this matrix, I'm basically choosing that maximum to be either coming from a match or from a gap or from a gap. So I'm choosing the scores of these 3 options, storing the best score that I could possibly get, and then reusing that to compute the next score here along with that and that and that. And then to compute the next score here along with that and that and that. And the next score here with that and that and that. Okay? So at every entry Mij, I have the best alignment score between the ith prefix of S1 versus the jth prefix of S2. And the best alignment is the best path through this matrix. So as I go through, I'm going to be remembering pointers up, diagonal, or left. And then the path through that matrix is basically going to say match, 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 insertion, mismatch, match, match, insertion, mismatch, deletion. Okay? And that's exactly the same operations that we had earlier. So when we transformed mouse into human, we had 1 insertion, 2 deletions, and a mismatch. That's exactly the same thing that we've been seeing all along. All right, so who feels that they're learning something cool so far? Awesome. So the answers are 195830. So this is very, very educational. Okay. So there's this duality. Every point in the matrix corresponds to the maximum score of that prefix to that prefix. And every path through the matrix corresponds to an optimal alignment, or sorry, corresponds to an alignment. And what we're going to be looking at is the optimal score by getting at the bottom right here. And then we're going to be computing and building up that alignment from the bottom to the top. Okay? So our steps are going to be first initializing the matrix, and then filling in the matrix, remembering the max pointers, finding the maximum score, and then tracing back the max pointers to build the alignment, and then returning the alignment of minimum length that has that score. Okay? So let's fill in this scoring matrix. So basically you go from the top row, you go down, and then every single time you have a choice, you basically remember the pointer that gave you that choice. Okay, so from here, the maximum came from that entry. And from here, the maximum also came from that entry. And here, the maximum came from either this entry or that entry. So basically, if this is a 0, I can get a 1 by either paying a penalty this way or a penalty that way. Okay? And then I compute the scores all the way down. And once I have my optimal score, I can just trace back the pointers all the way up. Okay? So let's look at an animation. We're going to first fill in this matrix this way, and then trace back the pointers that way. You guys ready? So, oops. So this is the trace back procedure. This is the score. And then this is the actual filling in. So start with 0 zeros, and then anything that's not a 0, I get added up. I get to the bottom, and then I trace back. And as I trace back, every choice of a pointer gives me a piece of an alignment. As I trace back, when I have a diagonal, I spit out 2 characters. When I only go in one direction, I spit out 1 character. And when I go diagonal, I spit out 2 characters at a time, okay? Because every time I move diagonally, I'm taking a character from each sequence. And every time I move either up or left, I'm basically taking only 1 character from one of the sequence and a gap from the other sequence. So we basically talked about how the update rule is actually local. It only looks at the neighboring cells. We compute the next alignment based on the previous alignment. And just like Fibonacci numbers, we had already the computation answer readily available when we needed it. And the table lookup avoided repeated computation. So we're computing the score of a cell from the smaller neighbors using the maximum. Remember, it's an optimization problem, so we're always taking a maximum. To compute the score of ij, I'm taking the maximum of 3 choices, either the score of the diagonal plus whatever score corresponds to the 2 characters that I have at hand, or the score from right above or from right to the left, paying a gap penalty each time. And there's only 3 possibilities for extending by 1 nucleotide: a gap in one species, a gap in the other, or a mismatch. And we're computing the scores for the prefixes of increasing length. We start with prefixes of length 1, extend by 1 each time until all prefixes have been computed. And when we reach the bottom right, the alignment score of the nth prefix of S1 and the nth prefix of S2 is basically the alignment of the full sequence S1 to the full sequence S2. And that's when we trace back to construct the optimal path to it by basically asking which of these 3 options gave me this maximum, okay? So if I have 13, I trace back the pointer, trace back, trace back, trace back, and this trace back gives me an alignment, because every time I move diagonally, I generate both characters, and every time I move horizontal or vertical, I generate 1 character. Okay? So we're computing all the alignment score from the bottom up. We're defining this prefix alignment score. We're filling up the table recursively, and we're expressing the alignment as a function of the previous entries. We only have 3 options, and it's only local computation. Okay? And then putting this into the 5 things that we needed to do before, what is our matrix parameterization? We basically have this mij that tells us about the alignment of the ith prefix and the jth prefix of S1 and S2. How do we make sure that the subproblem is finite? Well, it's just n squared, it's quadratic, which is polynomial, not exponential. We choose the traversal order. Well, I can go this way, or I can go that way, or I can go that way, as long as the 3 entries above, left, and diagonal up are always available when I need to compute. And then the recursion formula was computing the maximum of these 3 choices. And I had to actually remember which of the 3 cells led to the maximum. So, okay. This is basically, all you need to know. And Remember earlier when I said, oh, and I also built Excel function that does this? So alignment is actually so super simple and so cool that you can just encode it in Excel spreadsheet. So you can basically have your score matrix here that basically tells you what is the penalty of aligning an A to an A G to a G, a C to a T, a T to a T. I can change these scores to basically be minus 0.5, for example, and then that changes my alignment accordingly. How? By basically taking character pairs and then saying, well, what is the alignment of those two? And then that's just looking up the score of the corresponding sequence and the score of the corresponding sequence in this matrix. So that's basically matching that character on the rows and matching that other character on the columns, and then finding the index of those two characters here. And that gives me the score there, which basically means that every time I have an A aligning to an A, I get a +1. And every time I have a C aligning to a G, I get a minus 0.5. Everybody with me on that? So that's the first matrix. The second matrix is basically asking, well, what is my current score? This is actually taking all of the scores and computing them all the way down. This is basically choosing the maximum of the entry to the left plus the corresponding gap score from here, or the entry to the right, to the top, plus the score here, the gap score, or the diagonal up there, plus whatever score that diagonal was giving. And that basically computes the score all the way down here. Notice in this alignment, I have TAAC matching, and then I can move along the diagonal here. I have another diagonal here of ATCT matching, and I can just move along the diagonal here. And nothing much matches here, so I just, kind of meander my way through here, and then eventually make it all the way down. And then I have another matrix that basically remembers the maximum pointers. And this one basically says, I'm gonna say, oh, the top was part of the maximum if that score actually matches whatever score I would get from the top. Or the left side is part of the maximum if the left side matches that sum up here. Or the diagonal is part of the maximum if that diagonal entry matches the sum that I would get from here. And then the last matrix is the coolest one. It basically says, well, let's now start at the bottom and then move up based on whether the entry is pointing up, pointing to the left, or pointing straight. Okay, so this is a little fun. I had a huge fun making it, and then you guys can sort of hopefully fully understand alignments by looking at that. Okay. Oh, and then I even built some matrices that allow me to construct the sequences by concatenation. So if you zoom way in here, these are actually building up the alignment of one sequence and the alignment of the other sequence as I trace back. And then I end up with the two sequences aligned over here. Anyway, I'm totally geeking out here, but who thinks this is kind of cool? Let's see. Awesome. You guys are so rewarding. I love that. So 227100. Thank you for the 0 yawns. I really appreciate that. Would have broken my heart. Okay, so enjoy this at home. This is something that you can look at afterwards. Okay, so we know how to compute the best path. It's simply the number at the bottom right. We need to remember where it came from, and that's when we follow the pointers. We basically say, well, to obtain that score, I had to move here, and I had to move there, and so on and so forth. And then I retrace the path through the matrix by remembering all the pointers. So the time needed to compute all the way to the bottom was order of m times n, because the computation was local each time. I just needed to choose one of 3 options, and that's order of 1. It's a constant operation. The space needed was order m times n, because I needed to remember the full set of all of the pointers to get all the way back. Okay? So We basically completed the lecture, but we have another 3 minutes technically. So we're going to cover some advanced topics. And as I promised, I'm going to run over just a few minutes. So advanced topics. Let's look at some variants of dynamic programming. The first one is, do we really need to have the full matrix computed? Remember, as I'm tracing back, I'm kind of sticking close to the diagonal. Because if I move too far this way, that's a lot of gaps. And then to get back, well, I need to pay for a lot of gaps as well. So maybe one idea might be to do bounded dynamic programming, where I only look at a radius surrounding this diagonal, and I kind of ignore everything else, okay? So one idea is to basically only look at k characters of the diagonal and ignore everything else. We've suddenly changed an algorithm from an order n squared algorithm to effectively n times 10, or whatever my window might be. So if I have 1,000 by 1,000 sequence, I basically have to only do 1,000 steps, well, maybe a window of 10 or 20 surrounding that. So I've basically taken an order n squared algorithm to an order nk algorithm, whatever that k might be, okay? Who's with me on this one? So notice this is no longer optimal. This is a heuristic. We might not have the optimal solution, but if there is a pretty good solution, we'll basically find it. If we don't find it, it's basically a crappy solution anyway, so we might not care. So 25, 6, 0, 1, 0. All right, the next extension is going to be linear space, okay? And now brace yourselves. This is totally awesomely cool. If I want to compute just the maximum score, do I need to remember all of the columns? The answer is no. If I just want the optimal score, all I need to do is compute the scores here and then use them to compute the scores there, and then throw the first one away. And then use those to compute the third column, and then throw the first 2 columns away. And then use the third column to compute the fourth column. And after I've computed the fourth column, I can just throw the first 3 columns away. Okay? Everybody with me on this one? So all I need to do is remember 1 column at a time as I'm computing the next one. So in terms of space, I can basically use just linear space and walk through my alignment in only 2 columns at a time. And then the other columns, I can just throw them away. So 18, 7, 4, 0, 0. Now, that's all nice and good, and I have my maximum score. If you've understood the lecture so far, now's a good time to turn off and just go watch TV, because it's about to get a little more complicated, okay? So brace yourselves. I've computed the score, but I can't trace back anymore. I'm stuck because I've thrown away all my other columns. Could I possibly do linear space alignment in still quadratic time with a traceback rather than just the score? So that's something that I could just let you think over at home, but here's what we're going to do. We're going to sketch the algorithm very, very quickly. So what about the pointers? Can I still remember the pointers? And the idea is awesomely cool. I can compute, using only 2 columns at a time, the best scores and the best pointers for this column here in the middle, right? And I've only had to compute the left half of the matrix. I can also compute the best outgoing paths from the right to the middle with the score and anything that gives me an optimal score here, because the algorithm is reversible. Instead of computing from the top to the bottom, I can just compute from the bottom to the top. Instead of computing all the alignments, the max scores for the prefixes, I can just compute the max scores for the suffixes. So I can run the exact symmetric algorithm to compute the maximum suffix score starting at that point and pointer starting at that point. And then I can just add up the two to basically find the best sum of scores starting at the top left and ending in this middle column, and ending at the bottom right, starting from anywhere in this middle column. Summing up these two will basically give me the maximum score somewhere in the sum of these two columns. And it won't just give me the maximum score, it will also tell me, and here's the pointer of how I exit, and here's the pointer for how I enter that column. Let's see, who's with me so far? So I've computed the forward algorithm from the left all the way to the middle, and then the backward algorithm from the end all the way to the middle. I've summed up the two scores. That gives me the maximum score where I'm traversing that middle, and the maximum and the entry point to enter that middle column and to exit that middle column. I love seeing your answers here, because it tells me that you're being honest when you tell me you're actually following. I don't expect everyone to follow here, and that's okay. So we have 12, 8, 8, 3, 0, which is just fine. So we now have the maximum score in the middle column and the best point of entry and point of exit. And how long did it take us to compute that? It took us n squared. And that's okay, because now that I have my entry and my exit, I can just recurse. And I can now, the second time around, ignore this entire space and ignore that entire space. Because I know that I never have to move down. I only move up and to the left. And I never have to move up. I only move down and to the right. So after I've computed the first time with order n squared, the second time, I only have half of that, because I only compute half the matrix now. So it's n squared plus half of n squared. And the next time around, it's half of that and half of this. So the total runtime is m times n plus half of that, plus a quarter of that, plus an eighth of that, et cetera, summing up to just 2, which is a constant. So I've basically done order mn computation with only linear space, computing all of the midpoints, and then tracing back and recursing, and then effectively recomputing my path, okay? The reason they call it the 4 Russians algorithm is because there's 4 Russian names, and this dates from 1970, and I think it's just a very cool algorithm. So what did we see today? We saw dynamic programming and the concept, the foundational concept of reuse of computation. We saw how we can order the subproblems and fill the table of subproblems results, and how we can read the table instead of repeating the work using the Fibonacci example. Then we saw sequence alignment using edit distance and the scoring functions, and built a dynamic programming matrix. And we saw how the matrix traversal path has this duality where it corresponds to an optimal alignment. And we basically looked at why comparative genomics is so important, how we can read functional regions through molecular evolution, how to translate a problem from biology to computer science, how to formulate it, why it's hard, why there's an exponential number of alignments. And then we built up dynamic programming using Fibonacci, how we can reuse the subproblem by ordering the computation and looking up in a table. And then the very basic recipe of choosing your parameterization, understanding your subproblem space is finite, choosing the traversal order so that all the results are available when you need them, and then the recursion formula and the traceback. And then we apply that to sequence alignment, how it's an additive score. We built up a solution from the smaller parts. We use this prefix matrix that gives us finite subproblems and explores an exponential number of paths using only their finite choices. Every single time we remember the maximum that gave us rise to the score. And when we got to the bottom right, we traced back to construct a path that gave us that optimal score. One of the paths that gives us this optimal score, and there might be multiple paths that give us this optimal score. And then there are multiple optimal alignments. So every path corresponds one-to-one to an alignment. And when we found an optimal path, we had an optimal alignment. And every entry corresponded to the maximum score for that prefix. And we also saw these very cool algorithms for linear time bounded DP, which is a heuristic, but linear space DP, dynamic programming, which is an optimal solution, but in linear time. Okay. One way to avoid allocating and freeing memory, which is usually very slow. Okay, that's not for me. So who feels that they've learned some cool stuff today? So let's see who feels that they learned something. Relaunch polling. Awesome. Very cool. So 235100. And then lastly, who feels that they're with me for the whole lecture, or at least for the first 3 parts of the lecture? Nice. Awesome. And then the last poll I'm going to do is how was the pace? So 226000. And then how's the pace so far? Let's see. Good. Bunch of just rights, a little bit of too fast, a little bit of too slow, which is what we would like. Awesome. Good. So we have 20 people smack in the middle, 6 above, and 2 below. Okay. So thank you guys. Again, thank you all for showing up. You know, and then looking forward to the mentoring session tomorrow. Office hours, 5 p.m. today. Recitation, 3 p.m. tomorrow, and then mentoring session, 4 p.m. tomorrow. Remember, record your self-introduction videos, fill out your profiles using the templates, and then upload your profiles and fill out the first day survey if you haven't done already. Links to all of those are from the Stellar page. Who feels that they've learned something cool today? Yes? Awesome. Good. All right. Thanks, everyone. See you on Tuesday. Have a nice weekend, and then see you tonight and tomorrow and all that.